Okay, we're going to be talking about improving soybeans for Tennessee and the Mid-South region. My name is Vince Pantalone and my PhD student. What we do is develop high-yielding varieties for producers in Tennessee and the Mid-South region. We incorporate plant resistance to soybean diseases or environmental stresses such as drought tolerance to protect high yields. We develop herbicide-resistant varieties for producers in Tennessee and the Mid-South region. And we develop value-added soybeans, for example, higher protein and oil combinations to satisfy the increased demand for 48% soybean meal protein. We develop high oleic and low linolenic lines, enabling zero trans fat soybean oil. We improve plant breeding methodologies, including the use of DNA technology for targeted genetic gains. And we investigate genetic control of important traits and apply that knowledge to soybean improvement. We train graduate students in traditional and modern plant breeding to become the next generation of plant breeders. We develop new conventional and glyphosate-resistant varieties. University of Tennessee Ag Research releases these new varieties to Tennessee Advanced Genetics. Tennessee Advanced Genetics sublicenses to Unisoft Genetics or other marketing companies. Farmers purchase seed from these marketing companies. Every bushel of genetic gain accomplished by our soybean breeding program results in increased income to soybean producers. Over the past 3 years, with an estimated state average yield of 47.5 bushels per acre, an approximate commodity price of $9.19 per bushel, the impact of our new variety development efforts was 85,877 acres multiplied by 47.5 bushels per acre multiplied by $9.19 per bushel equals $37.5 million in revenue directly to soybean producers. Ag economists estimate that soybean commodity production results in about a 9-fold increase in revenue due to soybean barge and train shipment, silo and container storage, oil crushing, and protein meal extraction plants and biodiesel processing plants. Thus, the $37.5 million in revenue generated by seed sales of our varieties and the productive performance of our varieties translates to $37.5 million times 9, or $337.5 million in realized economic impact for the region for the past 3 years. I want to mention a couple of our new varieties that we just released over the past year. The first is TN16510R1, a new variety for commercialization. It's glyphosate resistant and it's off patent, so the seed price, the cost that you would pay for the sale of the seed is much lower than the Extend varieties. It has resistance to stem canker, resistance to southern root knot nematode, resistance to peanut root knot nematode. It's got white flowers and gray pubescence, a determinate growth habit, and a relative maturity of about 5.0. The yield in the 2018 USDA Southern Uniform Test across 17 environments showed 58.4 bushels per acre for TN16510R1 compared to the checks at 56.3 bushels per acre. The next variety, TN15507, is a new conventional variety for commercialization. It has high protein meal, about 50.5% protein meal, has resistance to stem canker, white flowers, gray pubescence, a determinate growth habit, and we actually have a project to change that to indeterminate, and a relative maturity of 4.7. In yield in the 2018 USDA Southern Uniform Test over 15 environments for the Maturity Group 4 late test, TN15507 yielded 56.8 bushels per acre compared to the checks at 54.8 bushels per acre, and those were very good checks, including Extend varieties. Next, we want to talk about hydrogenation of soybean oil. It improves shelf life and stability. The trans arrangement increases the melting point, shelf life, and flavor stability of the hydrogenated oil. Because of these technical properties, partially hydrogenated oils have been used in the food industry in such products as margarine, shortening, and baked goods. But there is a significant loss of domestic market for soybean oil because based on new scientific evidence, FDA has determined that partially hydrogenated oils, which are the primary dietary source of trans fat, are not generally recognized as safe or GRAS in any use in food. In June 2015, the FDA issued its final ruling that partially hydrogenated oils are banned in the United States. This is due to health risks associated with the consumption of trans fat, and therefore food manufacturers would no longer be permitted to sell partially hydrogenated oils either directly or as ingredients in another food product. New Tennessee high oleic lines with natural gene mutations requires 2 recessive genes. There are 2 additional genes for low linolenic acid that we're working on incorporating in our new high oleic soybeans. The Tennessee high oleic lines exceed the Monsanto transgene. We easily get over 80% oleic acid. It's equivalent to or greater than the DuPont Pioneer transgene, and it's equivalent to extra virgin olive oil. My graduate student Lauren Richardson published a paper in January of 2020, which became the article of the month for the Journal of the American Oil Chemists Society. Our next variety release will be a high-yielding, high oleic, low linolenic line. It has 4 recessive genes, 2 FAD2-1A and FAD2-1B, the FAD3A and the FAD3C, as omega-6 and omega-3 desaturase genes. It provides superior oxidative stability for food products, fuel, and lubricants. There's no hydrogenation in oil, and so there's no trans fat. The yield in the 2019 USDA Southern Uniform Test, prelim test, over 6 environments across 5 states for the maturity group 5 early, show that TN18 4110, which is actually a 4.9 in maturity, yielded 59.3 bushels per acre compared to the high yield checks, which were 54.4 bushels per acre. So we think we have a new variety on our. We greatly appreciate your support for our soybean breeding and genetics program. We do a lot to keep soybeans as valued commodities in the soybean business. And next, my graduate student, Chris Wyman, will talk about his research with soybean cyst nematode. Thank you very much.
Heteroduroglycines, commonly known as the soybean cyst nematode, is an obligate endoparasitic nematode, meaning that the nematode must enter the host plant in order to feed. The soybean cyst nematode was first observed in Japan in 1915, but it wasn't until 1954 that it was first reported in the United States in North Carolina. SCN remains to be the most economically important pest of soybean today, resulting in an estimated $1.5 billion loss each year in soybean production in the United States. The soybean cyst nematode eggs overwinter within what is called a cyst. The SCN cyst is lemon-shaped and brown to tan in color. In response to molecular signals released from the soybean root as they begin to grow, SCN juveniles hatch and make their way to the root. SCN juveniles then penetrate the root using their needle-like mouthpart called a stylet with the aid of enzymes and other proteins that the nematode produces. The juvenile nematode will travel through the root and excrete enzymes and effector proteins to form a feeding site, which is called a syncytium. The syncytium is formed from the fusion of multiple root cells and are only fully formed in susceptible soybean lines. Once the SCN nematodes develop to adulthood, the male stops feeding and exits the root. The female continues to feed and swells to a side, which ultimately causes the body to exit the root. Once the female body protrudes from the root, males and females reproduce. SCN females can then produce roughly 600 eggs, the majority of which remain in the female, which becomes the cyst once the female dies. This life cycle can take place in as few as 22 days under ideal conditions, and multiple life cycles can occur in a single growing season. The map to the right depicts the distribution of SCN in the United States, where we can see that soybean cyst nematode inhabits all major soybean producing areas. Where some nematicides and other management options are currently used to limit loss due to SCN, the most productive practice have been the use of crop rotations between soybean and non-host crops and the planting of soybean lines that are resistant to SCN. Currently, however, the vast majority of lines resistant to SCN are derived from the PI 88788 source of resistance, which uses a high copy number of the RHG1 resistant locus. Some other resistant lines have been derived from the Peking and Hartwig sources of resistance, utilizing the high copy number of the RHG4 resistant locus. It is important that more lines become available with differing sources of resistance to SCN in an attempt to keep the parasitic nematode from overcoming host resistance and devastating soybean production. The focus of the research I would like to present today was to test candidate genes that may also confer resistance to soybean cyst nematode. 10 candidate resistance genes were selected based off of a difference in the epigenetic methyl tags of resistant and susceptible near isogenic lines. Epigenetic markers or methyl tags are found on DNA that either promote or inhibit gene expression, which would further denote differences in the two different near isogenic lines. Each of the selected 10 genes had methylation tags on either the susceptible line or the resistant line, but were not found on both. In order to characterize these 10 candidate genes for resistance to SCN, each were individually incorporated into plasmid DNA and then transformed, or in other words inserted, into agrobacteria. The agrobacteria was then injected into soybean plants just below the cotyledon, which after 2 weeks produced new roots at that location. These newly formed roots are known as hairy roots. These hairy roots should overexpress the candidate gene of interest. To determine if we indeed successfully overexpress the candidate gene, we check to see if green fluorescent protein was expressed in the root. This green fluorescence protein was also in the plasmid we utilized. So hairy roots that fluoresced under the fluorescent microscope were thought to also overexpress the candidate gene and were ultimately used to test the candidate genes for resistance to soybean cyst nematode. The original roots of the selected plants were cut off, and the hairy roots, overexpressing the gene of interest, were planted in the growth chamber. Each plant was then inoculated with SCN race 3, and SCN cysts were counted 5 weeks later. We used a negative control of the susceptible near isogenic line that was injected with agrobacterium without any added genes other than an empty plasmid for comparisons. When comparing the candidate genes to the susceptible near isogenic line, 4 of the 10 genes were identified to have significant lower SCN cyst counts, 3 of which had an average of less than a quarter the amount of cysts as the negative control. The results of this study show 4 candidate genes, when overexpressed, conferred significant resistance to soybean cyst nematode race 3, which may be beneficial in developing future lines resistant to this pest. These 4 genes are currently being tested for resistance to the root knot nematode, and further assays will test the overexpression of these genes against SCN race 2. In the future, we hope to design molecular markers associated with the active form of these 4 genes that in turn can be utilized in breeding efforts to produce new soybean lines with a resistant to soybean cyst nematode. More studies may focus on plant growth and defense trade-offs associated with the expression of these genes, as well as the effects of stacking new resistant genes with those already commonly utilized, such as RHG1 and RHG4. These 4 novel resistant genes may further improve our breeding efforts when working to counteract the negative economic impact of the soybean cyst nematode.